Hello, everyone. Today, we will introduce peptide modification. There are 4 parts: overview of peptide modification, available modifications, application, our services. Peptide modification refers to the covalent and generally enzymatic modification of proteins following protein biosynthesis. Proteins are synthesized by ribosomes translating mRNA into polypeptide chains, which may then undergo peptide modification to form the mature protein product. Chemically synthesized peptides have free N-termini and C-termini. The modification is necessary for research and application of peptide. Common peptide modifications include following but not limit this: N-terminal modification, C-terminal modification, unusual and non-natural amino acids modification, peptide PEGylation, fluorescent dye/Fret pairs modification, post-translational modification, cyclic peptides, peptide conjugation, custom labeling, peptide tags. Via N-terminal acetylation, the charge is removed from the amino terminus of a peptide. Generally, acetyl modification is recommended to make the peptide more closely mimic the charge state in the native protein. Include following: acetyl, formyl, pyroglutamic, P-glu, fatty acids, urea, carbamate, sulfonamide, alkylamine. The default C-terminus of a peptide is either a free carboxylic acid or an amide. C-terminal amidation removes the charge from the C-terminus of a peptide. This may reduce the overall solubility of the peptide. Include amides, N-alkyl-amides, aldehydes, esters, p-nitroanilide, PNA, 7-amino-4-methylcoumarin. Apart from the 20 natural L-amino acids, there is a multitude of non-natural or unusual amino acids available that can be built into synthetic peptides. There are many different reasons to incorporate non-natural amino acids, such as to enhance affinity, selectivity of stability of peptide drug leads. Another application is the use of non-natural amino acids for induction, or stabilization of secondary structures. PEGylation is the process of covalently attaching polyethylene glycol, PEG, chains to peptides, proteins, or other biomolecules. PEGs are polymers that are non-ionic, non-toxic, biocompatible and highly hydrophilic. For PEG peptides, the PEG is usually attached to the peptide using standard amide bond formation, but other linkage chemistry are possible as well. Fluorescently labeled peptides are frequently used for in vivo biomedical imaging, protein binding and localization studies. Fluorochrome conjugated peptides can be visualized by fluorescence microscopy or other fluorescence visualization techniques. Most frequently, they are incorporated at the N-terminal end, or at the side chain of a cysteine, cis, or lysine, lys, residue, for example at the C-terminal end. Post-translational modifications, PTMs such as protein phosphorylation, protein methylation or protein acetylation play an important role in virtually all biological processes. Therefore, the synthesis of post-translationally modified peptides, for example phosphopeptides, or sulfated peptides, acetylated peptides, methylated peptides, peptides with citrulline, or peptide glycosylation, is crucial for many projects. There are also other peptide modification, such as cyclic peptides, custom labeling, custom conjugation, peptide tags. The multiple modification of peptides can reduce the effect of charged C or N-termini during ELISA binding. Amidation not only enhances the activity of peptide hormones, but also prolongs their shelf life. This increases their ability to enter cells, alter structure to better understand biological function, improve overall peptide stability, or enhance immunogenicity for antibody development and production. Created Peptides provides a variety of post-translational modified peptides for proteomics and epigenetics. In addition, we will make appropriate modifications to the peptide according to our customers' needs. Created Peptides is specialized in the peptide products and services, providing a confidential and efficient service at competitive prices. Every step of peptide synthesis is subject to Creative Peptides' stringent quality control.